Hello again, students. Um, I'm actually recording night time. Yeah, this is still on a Sunday, so it's still night time. So this is a recorded lesson, which will be shared to you the link for tomorrow's lesson. Okay. So as far as the syllabus is concerned, it's just. So as far as the syllabus is concerned, we have actually done until cholinergic synapse. Okay. So today's lesson is going to look at the neuromuscular junction, objective number 10, as well as if we have time, we will look at the ultra structure of striated muscle. Okay, if we have the time. Okay, so let's read. Describe the role of neuromuscular junctions, which I have already mentioned a little bit. Yeah, the T-tubule system and the sarcoplasmic reticulum in stimulating contraction in striated muscle. And then I'm going to look at describe the ultra structure of striated muscle. Ultra structure meaning in detailed, with reference to sarcomere structure using electron micrographs and diagrams. Okay, so we tried to cover objective number 10 as well as objective number 11. Okay, so I did mention about the neuromuscular junction, meaning the motor neuron will eventually terminate, at an effector. And normally the effector is muscle. It can be a muscle or it can be a gland. Okay, so this is definitely motor neuron here. This is a motor neuron. Okay, motor neuron. And if it terminates at a muscle, and like this one here, this is a muscle, then this is called the neuromuscular junction. Okay, neuromuscular junction. Of course, the arrival of this electrical impulse or nerve impulse at the neuromuscular junction will cause this muscle to contract. Okay, cause this muscle to contract. Okay, so let's just have a look at the structures of the neuromuscular junction. So it looks familiar. For example, the presynaptic membrane, the vesicle which contains acetylcholine, the chondria, the myelin shield, the synaptic cleft or the synaptic gap. The only perhaps one of the differences, or one of the main differences, is the membrane here, the postsynaptic membrane, okay, which is called the sarcolemma. Okay, because it's no longer a postsynaptic neuron, but it's called postsynaptic membrane, which is the muscle. The covering of the muscle is called the sarcolemma, the outer covering, which can be called the membrane. The membrane of the muscle, the cell membrane of the muscle is called sarcolemma. Okay. So if you read this text, neuromuscular junction, specialized form of synapse found between motor neuron and skeletal muscle fibers. It allows motor neurons to stimulate muscle contraction. Both the membrane at the end of the motor neuron and the sarcolemma membrane of the postsynaptic membrane, the muscle, are highly folded. Other than this, both the structure and the way it functions are very similar to a normal excitatory synapse, meaning the cholinergic synapse. Okay, cholinergic synapse. The end of the axon contains vesicle, contain many vesicles which contain neurotransmitter acetylcholine and many mitochondria. The arrival of nerve impulse at the end of the axon causes the same sequence as described earlier for a synapse Eventually, the entry of sodium in the sarcolemma causes a change in potential difference at the end plate potential. If this reaches threshold, action potential is formed, which spreads to the muscle, causing muscle contraction. Okay, so basically, the mechanism is the same. Okay, the mechanism is the same. What do I mean by the same? Is we skip the muscle contraction part, yeah, because the muscle contraction part has its own mechanism, but the way the action potential is generated in the muscle is the same, yeah. Arrival of nerve impulse causes calcium channels to open, so calcium will enter, okay, and it causes the vesicle to fuse with this vesicle contain acetylcholine fused with the presynaptic membrane. Acetylcholine is released into the synaptic cleft. Acetylcholine binds the receptor. It causes sodium channels to open, and then action potential occurs in the muscle. Okay, so it's up to that. If it's up to that point, from that point onwards, it will be slightly different because now we're going to talk about later on how the muscles here contract. Okay, so that detail will be mentioned later on, not today, but perhaps on Tuesday. Okay, so here, as you can see, this is the microfibril. We'll talk about this microfibril. Where does this microfibril come from? And the sarcomere, which is the structural unit of microfibril. We're going to talk about that later on. Okay, and then just to introduce to you, new terms like sarcolemma, okay, which is already the membrane of the muscle, and then the T-tubule system. The T-tubule, yeah? The T-tubule is here. Okay, it's equally important, which we will look at that later on, and just some images, beautiful images. You can see here. This is transmission electron micrograph. Okay, transmission electron micrograph. So transmission electron micrograph is a very detailed image of the muscle fiber and the nerve and the neuromuscular junction. Okay, all right, and let's now look at how does the arrival of nerve impulse at the neuromuscular junction cause the muscle fiber to contract. Okay, so we will look at the contraction part in detail on Tuesday. We will look at the sliding filament model, yeah, of muscle contraction. So this will be mentioned and taught on Tuesday. Yeah, now this is a very interesting image here. As you can see, this is the motor neuron. Yeah, this is the motor neuron, yellow in color, and the pink color here is the muscle. Okay, muscle fiber. Okay, now let's have a look at this one here, the events. This is important. Yeah. Now, if we look at number 1 until number 6, okay. If we look at number 1 until number 6, okay, 1, 2, 3, 4, 5, 6. Yeah, these are all the same, just like in synaptic transmission. Okay, between motor neurons, or between sensory neuron, or between motor and relay neuron, or between relay and between sensory and relay neuron, or between relay and motor neuron. These are all the same. See, action potential arrive. The arrival of action potential causes calcium channels to open. Calcium will enter. The vesicle containing acetylcholine will fuse with the presynaptic membrane. Acetylcholine is released by exocytosis, binds the receptor on the postsynaptic membrane or sarcolemma. Okay, so that is the only thing that changes now, sarcolemma, and then sodium ions will enter. Okay, sodium channels will open, and then sodium ions will enter. So that will make the inside positive, and then the outside negative. okay, and then this one will go down, okay, will go down, will go down, will go down until it reaches this muscle. This is the muscle here, the myofibril, which causes the contraction. Okay, so like I said, 1, 2, 3, 4, 5, 6, they are the same. Okay, these are the same. The only difference is the sarcolemma as the postsynaptic membrane. So the name of this postsynaptic membrane is no longer postsynaptic neuron. Yeah, this is not a neuron, but this is a muscle. So this is also called postsynaptic membrane, but specifically it's called sarcolemma. So from number 1 to number 6, these are the same. Okay, so events at the neuromuscular junction are the same, but then events in the muscle fiber, this is different. Yeah, this is something not different but additional, because now you will come across new terms like T-tubules. Okay, and then you come across this troponin, tropomyosin, myosin. Yeah, so what happens here in 7, 8, and 9 is muscular contraction. Okay, this the events that causes muscles to contract, and this one will be covered. Yeah, this one will be covered on Tuesday. Yeah, how muscles contract. Okay, of course it comes from initially the arrival of action potential. Okay, and of course followed by the role of the calcium. The binding of the calcium causes muscle contraction. So that one, just keep this. What I'm trying to say is keep this in mind. Okay, this one will be separate on Tuesday on muscle contraction. In other words, we're going to look at the sliding filament model. So in order to understand muscle contraction, we will have to look at the sliding filament model. Okay, this is taken from a different textbook. Okay, as you can see, so the arrival of nerve impulse here causes the inside to become positive, yeah, outside negative, and then this one will go down. Okay, like that. And of course, the previously positive will become negative. This one later on will become negative, and then positive, and then become negative again, and positive. So it will have to go back to its resting potential. Okay, so that one you should know already. Yeah. Okay. So when action potential travel down this T-tubule, yeah, this T-tubule, we'll have a look at the T-tubule later on. What happens is that calcium will be released by this sarcoplasmic reticulum. So basically, this is sarcoplasmic reticulum. Now, sarcoplasmic reticulum is none other than endoplasmic reticulum. Yeah, this is basically endoplasmic reticulum. The endoplasmic reticulum in the muscle is called sarcoplasmic reticulum. So it's just given a special name. Yeah. So the endoplasmic reticulum in muscles is called sarcoplasmic reticulum, and this is the one that contains calcium. Now, what is important here is that when nerve impulse travel down this T-tubule, okay, or I can put down here action potential travel down this T-tubule, so calcium will be released from the will be released, yeah, from the endoplasmic reticulum or sarcoplasmic reticulum, and this calcium will bind to the muscle. Okay, In other words, will bind to troponin, yeah, will bind to troponin. If we just want to double confirm on that, the sliding filament model, as you can see here, calcium will bind to troponin. Okay, so this is the sliding filament model, which we will cover on Tuesday. Yeah, so don't worry about that. Most important thing is you need to understand first the earlier information. Okay, so calcium, the arrival of the transmission of or propagation of action potential down the T tubule will cause the release of this calcium from the sarcoplasmic reticulum. Then the calcium will bind to troponin on the muscle. So this calcium will bind to troponin. Okay, we'll talk about this on Tuesday, and causes muscle contraction. So this is the part that we will learn in detail. This part here is the one that we will learn in detail on Tuesday. This one here is already done. Yeah, this is already done because it's the same. It's the same as in synaptic transmission. Okay, so as you can see here again, so you can see the minus sign there. That's because inside is already positive. Yeah, positive, 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 positive. Okay, whereas here there is no action potential here, so outside negative, inside, outside positive, inside negative. Okay, but the action potential comes from here. Yeah, and so the inside will become positive, outside negative. So it's like that. Okay, so when action potential spread along a muscle fiber, it is carried into the fiber along T-tubule membrane. This causes the release of calcium ions from the cisternae of endoplasmic reticulum. You see here it still uses the term endoplasmic reticulum or sarco, yeah, sarcoplasmic reticulum is the same. Okay, all right, so that's the part of the neuromuscular junction. Yeah, now let's look at the different types of muscles. So when we look at muscles, there are actually 3 types of muscle. Okay, first one here, which I already circled just now, striated muscle, and then cardiac muscle, and then smooth muscle. So there are 3 different types of muscle. Okay, striated, cardiac, and smooth muscle. Both striated and cardiac muscles are it says here where's the term here, striated, yeah, looking for the term striated here. Okay, so smooth muscle has no striation, whereas cardiac and striated muscle, this one have striations. Striation means stripes, yeah, stripes. Striations means stripe. You know, like Adidas. You know, when you wear Adidas shirt, you have the black and white, and then black, white, black, white, black, white, black, white, and then black. So this is called striation, stripes. Okay, so cardiac and cardiac muscle and striated muscles, they both have striations or stripes, whereas smooth muscle, they don't have stripes. Okay, so these are just introduction to learn the different types of muscle. Okay, and then the striated muscle, this one will rely on neurons in order to function, whereas cardiac muscles, you can use a different color here. Cardiac muscles is myogenic, yeah, myogenic. Myogenic means it can contract on its own, it's automatic. Whereas striated muscle, it relies on neurons, the transmission of nerve impulse. Now, smooth muscle can be both, yeah. Whereas smooth muscle, it can be both. Smooth muscle may rely on uh, um, neurons, or it may not rely on neurons. For example, in arteries, when blood flows in them, in the lumen of arteries, the smooth muscle contracts and relax on its own, so it doesn't require nervous transmission. So it can be both, requires neuron transmission, or no need neuron transmission. That's for smooth muscle. Okay, so that is just a brief description on the different types of muscle. Okay, so you can see here for smooth muscle there is no striation. Striated and cardiac have striations. Okay, so that's the most important thing. Striated is neurogenic, cardiac is myogenic, meaning on its own it contracts and relaxes on its own. Smooth muscle is neurogenic and also not neurogenic. Yeah, both neurogenic and not neurogenic for smooth muscle. Okay, now let's have a look at this part here. So there's a bone here. What connects bone to a muscle is called tendon. Yeah? So tendon, that's the tendon here that connects the bone. Yeah, connects the bone with the muscle. Okay, it's called tendon. Okay, and here is what we see, a muscle. Yeah, this is a muscle here, big chunk of muscle here, and it consists of lots of muscle fiber. So a muscle has many muscle fibers, and each muscle fiber has many myofibril. Okay, so let's have a look at this one here. This is a good one. So here we have a muscle, okay, which consists of many muscle fiber. So here you can see this is one muscle fiber. This is a muscle fiber, one muscle fiber, and of course you can see it has many muscle fiber here, and each muscle fiber has many myofibril. You can see there. This is another one myofibril, another one myofibril, another, many myofibril. This is one muscle fiber, has many myofibril. Okay, so if we were to create like a flow chart, okay, so a muscle has many muscle fiber. It can be thousands of muscle fiber, and each muscle fiber consists of many myofibrils. Okay, myofibrils. Okay, so here, as you can see, this is a muscle fiber. So this is a muscle fiber. So if we were to zoom in this muscle fiber, this one has many myofibril. Many myofibrils, lots of myofibril in each muscle fiber, and this whole thing here is called muscle chunk. Yeah, big muscle chunk. Right, here's a very good image of that. You can see one muscle fiber consists of many myofibrils. Okay, so what you see here is only one myofibril. There's another myofibril, another myofibril, another myofibril. This one is another myofibril, and so on. Okay. So this is very, very important. Yeah, this is very, very important. So when we look at a myofibril, this is a myofibril. So the structural unit, yeah, a structural unit of a of a myofibril is called sarcomere. Yeah, so this is one sarcomere, another sarcomere. Okay, so from here to here is one sarcomere, another sarcomere, another sarcomere, another sarcomere, and so on and so on and so on. Yeah, so sarcomere is the structural unit. Structural unit, you know, just like the filtering unit for the kidneys is called nephron. That's the filtering unit. For muscle or in the myofibril, it's called sarcomere. This is the structural unit. And we know about sarcolemma. Sarcolemma is the membrane, yeah? Membrane or the covering. It says here the fine transparent tubular shield which envelopes mean cover the fibers of skeletal muscle. So simply we call it the membrane, yeah, or the cell membrane of muscle. It's called sarcolemma. Okay, as you can see from here, sarcolemma. See the covering, the outer covering, yeah. And then you can see the sarcoplasmic reticulum. This is the endoplasmic reticulum, remember, okay, contains calcium, okay. So this is the T tubules, here, yeah, which is also the T tubules here going down, okay. So normally what happens is that when action potential occurs here, okay, going down here, okay, and that's when this sarcoplasmic reticulum will release its calcium. Yeah, and now the calcium will, this calcium here will bind to the troponin on the muscle, and that will cause a reaction. Of course, it's not as simple as you may think. You have to, again, you need to look at the sliding filament model, yeah, sliding filament model of muscle contraction. Okay, right. So here muscle fiber, this is one myofibril, another myofibril, yeah. Okay, as you can see, this is taken from another textbook. Okay, so remember the T tubules here, and then the chondria. So okay, so the membrane of sarcoplasmic reticulum have huge numbers of protein pump. This is very important point, yeah. Protein pumped, pump protein for calcium, that transport calcium into the cisternae of the sarcoplasmic reticulum. So what happens here, if I can just rephrase, yeah. So when action potential travels down, when action potential travels down this T tubule, okay, calcium will be released. Now this release of calcium is by facilitated diffusion, okay, facilitated diffusion. But then after the muscle contraction, okay, once the muscle contraction is completed, this calcium will re-enter the sarcoplasmic reticulum by active transport, yeah. So initially it's by facilitated diffusion, okay, facilitated diffusion, but then after that, after the muscle contraction, the calcium will re-enter, the calcium will re-enter the sarcoplasmic reticulum, and this is by active transport. Okay, active transport. And this is the reason why you will see lots of mitochondria. Yeah, you will see lots of mitochondria. And this is the reason why I want to explain that. One of the purpose of this mitochondria is for the pumping of calcium back into this sarcoplasmic reticulum after muscular contraction. Okay, that's a very, very important point, and that's why there's a huge number, it says here, there is huge number of pump protein for calcium. Okay, right, now let's look at, so here again, as you can see, this is one myofibril, yeah, one myofibril, another myofibril, another myofibril, another myofibril. Okay. Okay, now let's look at, there is a mistake here in the notes, yeah, in the, even the this is not the H band, yeah, this part here is not the H band, so there's a mistake, so I'm just gonna inform you that that is a mistake. Okay. Right. The rest is the same. So this is one structural unit, yeah, one structural unit for muscle, structural unit of muscle in myofibril. Sarcomere. Now, sarcomere starts from Z disc to the next Z disc. Okay. There is a line here which they call the Z disc. There's also another line here, Z disc. Okay, forget about the band first. Let's look at the Z disc. There's also an M disc. This is the M disc in the middle here. This is the M disc. Okay, it's very easy to memorize. M stands for middle. The M line, yeah, the M line or the Z line. Here, from different textbook, this is the M line, which I think it's middle, yeah, and this is the Z line. So between one Z line to the next Z line, Z line, this is sarcomere. Okay, sarcomere. This is another sarcomere. Okay, right. Let's clear that one. And then now you need to learn the three bands, the A band, the H band, and the I band, AHI, yeah? Now, the A band consists of myosin. This thick line here is called myosin. And the thin line is called actin. These are all proteins, yeah? Okay, so the A band contains myosin and actin. Okay, the I band, which is only from here to here, okay, only contains actin. This is only actin. This is actin, yeah? Actin. Whereas the A band contains both, yeah? Both actin and myosin. That's the A band. Okay. Now the H band, on the other hand, the H band, which is only this middle part here, can use the highlighter. Yep, this is the A band. Okay, this is only the H band. The H band only has myosin. Yeah, myosin. Okay, so that is something that you need to memorize. Okay, you have to memorize. There's no other way. Yeah, you have got to memorize. So if we look at again the M line, okay, this is the M line. Okay, M line. This is the A band. The A band starts from here. Okay. So from here to here is the A band. A band. Okay. So you can see the A band has the thick and the thin. Thick is myosin, the thin is the actin. Okay. And then the H band, something that the H band is this part here. Okay, this is the H band. The H band is only myosin. The I band, which is this one here, not really complete here. It's not wide enough. So this is the I band. And this is only actin. Okay, the thinner type of protein. Yeah, and then this is the thick one. Okay, so remember sarcomere is from one Z line to the next Z line. This is Z line to the next Z line. Z, Z, Z line to the next Z line. Okay, so this is one sarcomere. Okay, one sarcomere. The A band, remember the A band is only myosin. Okay. The I band is only actin. Okay. The H band is not shown here. The H band here is not properly shown. The H band is somewhere. It should be somewhere here. Okay, the H band. The A band is here. Is there here? Yep, this is the A band. It's not really clear, yeah? It's not really properly shown, the A band. But remember, the A band has both the thick and the thin, myosin and actin. Okay? Okay. So there's a way to memorize this. It says here, I band light. So I band means only the I band is only the actin. The A band is the myosin plus actin. Okay. All right. Now this is another one here. So remember, if sometimes you will be asked to label, yeah. So let's label this properly. As you can see here, this is the Z line. Z line, okay. Z line. This is the M line. Okay, M line. So this is, if you use a different color, this is the A band, okay, and then let's refer back to here. Yep, this is the I band. Okay, and this part here, just going to use a highlighter here, is the H band. Okay, this is the H band. H. Yep, this is the H band. Okay, so it's good to, so the H band, as you can see, the A band has the myosin and actin. The H band only has the myosin. The I band only has the actin. Yeah, so you have to learn how to actually label it. Yeah, so what happens during contraction? Okay, so when you can see during the contraction, you can see the, why do we need to label this I band? Yeah, so first of all, if I can use a color here, this is the I band. Yeah, so you can see that the during muscle contraction, the I band narrow down. Okay, the I band will become narrow, and then the A band, yeah, the A band remains constant. This is the A band. This one remains constant during muscle contraction. This is the A band. Constant. And then the H band. The H band this is the H band here, which is only the myosin. Yeah, you can see also shorten. Yeah, shorten. Okay, so let's see that one here very clearly. Okay, so it's good to start from, by not referring. It's good to actually do it without referring, and then later on confirming it by looking at the sample of answers. Yeah, so like I said, this is before contraction is like this. After contraction, the I band gets narrow. The A band, the blue color here, the A band constant. The H band becomes narrow. Okay, as you can see here. Yeah, so you can see the H band or H zone becomes narrower. The I band becomes narrower. Okay, the A band remains constant. This is the A band. You see, remains the same. Constant, the same. Okay, now as you can see here again, okay, you see the A band remains the same. the I band narrow, the H band becomes narrow. Okay, as you can see here, the H band narrow, the I band becomes narrow, the A band remains constant. Okay, so you will be asked in the exam on this. Yeah, so if you see here, the I band narrows, which is this one, the sarcomere shortens. Yeah, this is the Z line, sarcomere between two Z line. Yeah, so you can see this is the between two Z line is the sarcomere. This is sarcomere, and now this is the new sarcomere after contraction. Okay, so you can see it narrows. The A band stays the same width. This is the same, the A band. The H zone or the H band narrows, and the Z line become closer, yeah? Because the sarcomere shortens. The brown color here, the Z line shortens. Okay. So, myosin anchored on the M line. Anchored means it clings on it. You can see here, this is the myosin anchored on the M line, and actin anchored on the Z line. Okay, actin anchored on the Z line. Okay, now this is a very important slide as well, where you can see the cross section. Sometimes you're given a cross section like this, and you are required to identify from which band, yeah, from which band you see this. So you have the thick and the thin. This is from the A band. You have the thick only, so the H band or H zone. You have the thin only here, so it's the I band. Okay, cross section. Yeah, A band, H zone, I band. So I don't know. It's up to you to. There are many. Probably you can design or design, or you can design your own way of memorizing. I just AHI. Yeah, I just call it AHI. Yeah, AHI. AHI. AHI means thick and thin filament, excluding the H band. Yeah, because the H band is also within the A band. For example, this one here, this is the A band, yeah, and then the A band includes the H zone, but then the thin and the thick is actually this area here. The thin and the thick for the A band is only this one here. The H zone only the thick here. Okay, so that's why I wrote down here the A band thick and thin filament, excluding the H band or the H zone. The H band thick filaments only, I band thin filament only. So make sure you know thick means myosin. Okay, the thin is actin. Okay, likewise the thick here is myosin, the thin is actin. Okay. So on Tuesday, we will look at the role of tropomyosin and troponin and calcium in ATP. Yeah. So I hope you understand this. It's very important. You know the ultra structure of the striated muscle, yeah? Particularly the sarcomere. Okay. And then of course the role of the neuromuscular junction. Right. That's all for today. We'll see you again on Tuesday.